digestive enzyme that was discovered in 1833 by Payne-Pearzaz after being isolated from barley malt. This enzyme aids in the breakdown of carbohydrates by breaking the bonds between sugar molecules and polysaccharides through a hydrolysis reaction. It can be found in animals, plants, and bacteria. However, beta and gamma amylase are not found in animal tissue. There are 3 classifications of amylase, which differ in mechanisms of hydrolyzing the polysaccharide bonds: alpha amylase, beta amylase, and gamma amylase. Alpha amylase is a calcium-dependent enzyme composed of alpha helices and beta sheets, which forms maltose, glucose, or other simple sugars from starch. The alpha amylase enzyme is produced by serous acinar cells in the parotoid and submandibular glands and has a pH of 6.7 to 7.0. Beta amylase reacts with 1,4-glycosidic bonds in starches and is responsible for ripening fruit by turning starch into maltose. In seeds, beta amylase is present before germination with a pH of 4 to 5. Gamma amylase reacts on amylose and amylopectin to yield glucose and is the most acidic with a pH of 3. The evolution of amylase was key for humans to be more adaptive to different food sources. Even in a single type, amylase still has several different conformations, as seen in the pictures below of human salivary and pancreatic amylase.
We're going to focus our discussion on alpha amylase. This is the structure of human salivary alpha amylase. The structure consists of 3 domains, A, B, and C, and consists of 1 calcium ion and 1 chloride ion to stabilize the active site. The pink structures represent alpha helices, while the orange structures represent beta sheets within the structure. Alpha amylase catalyzes the hydrolysis of starch to glucose. This is why bread becomes sweet when chewing, because the starch is being transformed into sugar. Alpha amylase is produced in both the pancreatic and salivary glands of humans. Fluctuations in amylase levels can be used to diagnose some medical conditions that affect the pancreas. This enzyme is also important in bread making and brewing of beer. Yeast feed on simple sugars, which are products of amylase reactions. Though yeast produce amylase naturally, brewers and bread makers may add extra amylase to their procedures to create more sugars faster, since yeast break down the starches too slowly naturally. Additionally, amylases are used in fabric and dishwater detergents to dissolve starches.
Alpha amylase's active site works on a 5-ring long section of starch. Aspartic acid 174 attacks carbon 1 of one section of this chain, and then the oxygen of the glycosidic linkage attacks the hydrogen of glutamic acid 200. This creates a linkage between aspartic acid 174 and carbon 1 of one part of the original starch molecule. The other part of the starch molecule, which has now been cleaved, now leaves the reaction. Glutamic acid 200 attacks a molecule of water, taking one of its hydrogens and leaving a reactive OH group. This OH group attacks carbon 1, breaking the bond between the starch molecule and aspartic acid 174.